literally be a billion of these individual cells, and each one of them might be doing something different than the other ones. So picking apart who does what in the environment is actually very, very difficult. And so that...
Oops.
We tackle that problem in a bunch of different ways.
Could you repeat that?
Go ahead.
Sorry, could you repeat
that? The part about how it's hard to pick apart exactly what a billion different microbial cells are doing in a single drop of seawater. And so we are just trying to find strategies for how to solve the problem and find the answers to those questions. And one of the really fun, I think fun new things that we're doing is that we're pulling out individual cells. And this is not what we'll have you do, but this is something that we're doing in my lab. We pull out an individual cell of a microbe and we sequence its entire genome just from one copy. And now we can do that, so we can get a lot of information about the genes that this organism has and the things that it might be doing from this. And so that's sort of my end of things, is looking at the microbes and how they function in the environment. And what Dr. Steen studies is more looking at the actual functions that they're doing directly.
That's right. So as Dr. Lloyd said, there's a lot of bacteria, and she was talking about the oceans. We're going to do some work in lakes and streams also, and so all of that is true for lakes and streams. There's a lot of bacteria in there, and the vast majority of them are supposed to be there. They're not bad for you or anything. And the thing that I want to know about, and a lot of other people want to know, is what these bacteria eat. So when you eat food, you put it in your mouth and it goes into your stomach, and your body breaks down the food inside your stomach. But of course, bacteria don't have a stomach because they're too small. So what they do is they produce what's called enzymes, which are chemicals that break down food, essentially, organic matter, organic compounds like proteins and carbohydrates and all the stuff that you see on the nutrition information label on your food. They break them down into small pieces, and then they transport those small pieces back into the cell across the cell membrane. We don't really know a whole lot about what kinds of enzymes, these are called extracellular enzymes because they're outside of the cell. So they're totally crucial, they're totally important to breaking down the food that bacteria eat, but we don't really know what they are or how they work. And that's the thing that I'm really excited about, and that's sort of the subject of the project that we're going to be doing together. We have recently discovered, if you read the scientific literature in the past 20 years or so, people who have looked at a class of enzymes called peptidases, which break down proteins in the environment, and they sort of talked about it like all peptidases are the same, and there's really only sort of one kind of peptidase in the environment, in rivers or the ocean or whatever. And some colleagues of mine and I have recently discovered that there's actually a lot of different kinds of peptidases, and they do different things, they work in different ways, and they break down different kinds of protein. And we really don't know very much about what kinds there are, or what they're doing exactly, or what places have one kind of peptidase and what places have another kind of peptidase. And that's very exciting for me because actually measuring the amount of peptidases, we call it the activity of peptidases, is really very easy. It's something that you don't have to go to school for a long time to learn how to do. It's something that we can figure out how to do with Mr. Murray in a day or two. So what we're going to do is get some substrates, some chemicals that we use to measure the activity of different kinds of peptidases, and just go around to a bunch of places where there's natural water.
Do you mean that's what they're going to be doing?
Yes, that's when I say we, I mean the two of us and you all.
And you guys.
And we're gonna measure the activity of these different kinds of peptidases. And I'm very excited about this because a lot of times in science, the way you move things forward, the way you make an advance, is to learn some really complicated technique and spend a long time in lab figuring out how to do it, and then you do a whole lot of work to make some sort of incremental step forward. And it just happens to be that this situation right now, that we've discovered that there's these different peptidases that exist, but we don't know much about them, which means that we can really, just by measuring these ones, which is an easy measurement to make, we can really do something that's really a scientific advance. Nobody knows what kinds of peptidases are active in streams and lakes and estuaries and coastal seawater. And so just by measuring those over the course of a week or so, we're going to make a really big advance, I think, that I'm hoping that we will publish in a real scientific journal and other scientists will be very excited about.
If it all goes well.
If everything goes according to plan. And of course in science, nothing ever goes exactly according to plan. You hope that it goes close enough to plan that you can still get a good result.
So do you guys want to ask some questions?
Yeah.
Taylor, what's your question? Yeah, you're looking over at the computer screen. Taylor, you got a question?
My name is Taylor Messer.
We may need to get Mr. Murray to repeat the question because his speaking voice is very good and the sound is so good.
Go ahead. Or just speak loud.
What is the significance of discovering concentrations of certain enzymes in the world's oceans?
Oh, we can hear you. Hold on.
We can't hear you.
We lost it.
Can you hear it now?
Go ahead.
Okay. The significance is that it tells us exactly what kind of food these bacteria are eating. And if we know what kind of food they're eating, then we can make predictions about how much of the food, the organic matter that's in the world, is going to be transformed into carbon dioxide and end up in the atmosphere, and how much of it is not.
And and the world is getting warmer.
That is true.
That's happening. So this is going to change. And if we understand the process, then we can better react to the changes.
Great, thank you. Next question.
Hi, my name is Wojciech Shuhei, and my question is, how do protein enzyme break down in the water?
Can we get Mr. Murray to repeat the question?
Sure, how does protein enzymes break down in water?
Say that again. How do they work in water?
How do they break down in water?
You mean the enzymes themselves?
Yes, that was the question.
So there's two different things. The enzymes are breaking down the organic matter, the food, the proteins, and
their proteins themselves.
It's confusing because they are proteins themselves and they themselves break down. The way they break down organic matter is that you can think of them as sort of like a little glove. They have a little pocket in the middle, and what happens is a protein molecule will fit into the pocket. They attract a protein molecule into the pocket, and it fits there, and then they have little pieces of the protein that sort of act like scissors to break a bond inside the protein, which breaks the protein into smaller parts. So that's the process of the peptidase, the enzyme, breaking down a protein. But the peptidases themselves break down. They're either broken down by other peptidases, or they can be broken down by sunlight, or just by kind of the action of they kind of randomly fall apart. So bacteria in the ocean need to be constantly making new peptidases to replace the peptidases that are being broken down in the environment.
Great, thanks very much. Next question. I'm just going to go across. Did you have one? You'll get to the. Do you have one or no? No? Next?
How did you get interested in your project?
We're having audio problems. I don't know if you can hear us.
Yeah, we can hear you. Yeah.
Beautiful. We're back. Well, go ahead, give it a try.
Okay. So just from the very get-go, I grew up near the ocean, and it was always there. And so I always had this concept of this grand thing that was much bigger than I was. And now I'm just totally curious about whatever mystery is there. So that's really it. It doesn't take any more than that to get me interested in this.